Hi everybody, my name's Porik, and this will be my mini presentation on the CRISPR-Cas9 mechanism. So what exactly is CRISPR? Well, CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, as previously described. So I'm going to be using the same operon sequence as Kaylee had used, and just as a reminder, in purple we have the Cas proteins, in white we have the leader sequence, and then the rest will be our CRISPR loci. So there's three types of CRISPR mechanisms, and they differ in their usage of Cas proteins and molecular pathways. But it's important to remember that all types use Cas1 and Cas2 proteins, and on top of that, the type relevant to gene editing is type 2, which utilizes a Cas9 system, and this is the one that we're going to be focusing on in this presentation. So how exactly does CRISPR work? So we begin by having a phage injecting its own DNA sequence into a bacterium. A Cas9 protein is produced along with tracer RNA, and these two form a complex in order to form a mature form of mature CRRNA. So CRRNA functions as a guide to the invading gene into the Cas9 system, where it will be cleaved and degraded. And the cleaving ends are upstream of a very important sequence called the PAM sequence, also known as the protospacer adjacent motif. And this will tell us exactly where the Cas9 protein will be aligning. Here's just a visual of what this PAM sequence will look like relative to our green CRISPR loci. And generally the PAM sequence has a code of NGG. So let's begin with our animations. So first we see a phage inject its genetic code into a bacterium. And we see here that this genetic code actually aligns with a CRISPR locus. Now, there's actually one more thing that I'd like to add on to the operon that we have here, and that's actually a very important sequence that codes for the tracer RNAs themselves. So when we do produce those tracer RNAs and the Cas proteins, the tracer RNA will align with the CRISPR locus associated with the viral gene, and so will the Cas9. This will form a complex which will eventually form a CRRNA. Right, so what we see here is the Cas9 protein has formed its complex along with the tracer RNA and the CRRNA. And the CRRNA is now base pairing with the viral gene. And this will be pulling it through the Cas9 protein and aligning it with those scissor-like enzymatic regions. Eventually, the target gene will be cleaved and degraded.